Morning, students. Well, today our topic of discussion is chemotherapy. Well, what is chemotherapy? Well, many of us immediately think the word chemotherapy reminds us of probably cancer, right? Cancer chemotherapy, patients who are undergoing chemo treatment, right? So many people think of the term chemotherapy relating to the specific modern drug treatments used to fight cancer. Actually, modern chemotherapy began in the early 1900s with the work of Paul Ehrlich. You can see the picture of Paul Ehrlich. He was a German medical doctor and researcher who is regarded as the father of immunology and chemotherapy. In fact, he hypothesized that certain dyes could selectively stain harmful bacteria and without harming the host cells, that is, the person who has consumed the dye. He referred to such compounds as magic bullets and coined the term chemotherapy as a general descriptor for chemical remedies targeted to selectively kill infectious cells. Now, this definition includes all antibiotics as well as the more recently developed chemotherapeutic agents targeted to selectively kill cancer cells. So, with that, what are we understanding? Chemotherapy is not just targeted towards cancer. Any chemical drug, if it is consumed by a human to get rid of any kind of illness or a disease, that is called as chemotherapy, right? Okay. Now, building on the Ehrlich's work, okay, Paul Ehrlich stated this chemotherapy definition. It was Gerald Domark. He was a medical doctor employed by a German dye manufacturer, made a breakthrough discovery by finding that a dye known as prontosal, dosed orally, was effective in curing life-threatening streptococci infections in humans. There's an interesting event which happened during Domark's life. In fact, Domark had a daughter who was just 8 years old, and this daughter of Domark had a serious infection, and the infection was not cured by the available medications that time. So Domark was helpless. As a helpless father, Domark had to go for any of the resort that he had to save his little daughter. Finally, Domark came to a conclusion that he was working with these dyes. In fact, he was employed by the dye manufacturer. He used the prontosal dye itself as a life-saving drug on his own daughter, right? The first patient was his own daughter, and this actually discovery led to an extremely productive period of discovery in antibiotics. In fact, Domark was awarded with a Nobel Prize in 1939. The prontosal discovery led to a group of drugs which were discovered later called as the sulfa drugs or the sulfonylamide drugs. The sulfa drugs were discovered in early 1900s and found to be effective antibacterial agents. It inhibits the formation of folic acid in bacteria. We all know that folic acid is an important coenzyme which has to be present for various nucleotide metabolic pathway. By blocking that pathway, we are blocking the growth of bacteria. So it is a bacteriostatic agent. It stops the growth of bacteria. It prevents its further growth, and sulfonylamide can be synthesized from aniline in 4 steps. So in our biochemistry discussion, we are looking at the biological part of it as well as the chemical part of it, that is, how is sulfonylamide synthesized. So looking at the reactions, we start with the parent molecule, that is benzene. Now benzene undergoes nitration to form nitrobenzene. The nitration mixture is a mixture of concentrated nitric acid and sulfuric acid with a temperature of around 50 degrees. We get nitrobenzene. Nitrobenzene, in the presence of strontium and hydrochloric acid, is converted to aniline. Aniline, in the next step, gets converted to acetanilide. So acetylation reaction is what we are seeing here. Acetanilide, in the next step, is converted to 4-acetamidobenzene sulfonyl chloride. 4-acetamidobenzene sulfonyl chloride with the influence of ammonia is then converted to 4-acetamidobenzene sulfonylamide. 4-acetamidobenzene sulfonylamide is now converted to sulfonylamide by the removal of the acetyl group. Of course, there is acid hydrolysis here, and we also use sodium carbonate. So that's how we synthesize sulfonylamide here. Now that we have understood how a sulfonylamide is synthesized, let us see the applications of sulfonylamide. I told you that it is a powerful antibacterial agent. So if it is antibacterial, what are the type of bacterial infections do it treat? We can use sulfonylamide in treating pneumonia, urinary tract infections, bacterial infections for burn patients, cerebral toxoplasmosis, conjunctivitis and other eye infections. Well, okay, now we move on to the next drug that we are going to take as an example to study, that is sodium thiopental, or it is also known as pentathiol sodium. So the trade name is sodium pentothal among others. It is a general anesthetic that acts quickly but only for a short time. Now we have introduced a word anesthetic. So what do we understand by the word anesthetic? Any chemical or any drug which is used to bring about a transient unconscious state, right? The person has to be put on an unconscious state for a limited period of time during surgeries or any kind of therapies. One advantage of this particular drug, sodium thiopental, is that it can be used in surgery even for pregnant mothers with no effect on the fetus. Usually other kind of, for example, chloroform is also a potent anesthetic that we use in surgeries, but when chloroform is used, it can bring about certain kind of harmful effects on the fetus. But this particular thiopental is safely used even on pregnant mothers. Now let's see the chemistry of it. Sodium pentothal is synthesized starting from thiourea, right? So sodium pentothal is formed by combining a complex ester and thiourea in a two-step process. The complex ester is a diethyl malonate. Please observe the structure here. So we have the two ethyl groups. It is diethyl malonate. So this is diethyl malonate plus thiourea. So this is thiourea. We know the structure of urea, NH2CONH2. Instead of oxygen, we have sulfur here. That's why it is called thiourea. So this diethyl malonate plus thiourea combines to form pentothal. So this is the pentothal. It is a structure that we get. This complex ester, that is diethyl 2-ethyl 2-malonate and thiourea, undergo nucleophilic acyl substitution to form pentothal. The sodium salt of pentothal is then formed by a hydrogen abstraction. So this is subjected to sodium hydroxide. The hydrogen is abstracted and that place sodium gets attached. So we get sodium pentothal. So sodium pentothal is the active drug that we have. Looking at the applications of sodium pentothal or thiopental, it is used in the induction phase of GA, that is general anesthesia. It is used to induce anesthesia in animals. So what are the consequences where we have anesthesia being induced in animals? In various animal studies. Okay, animal studies, particularly in clinical trials, we do that. To induce medical coma, to terminate the seizures, for the purpose of euthanasia. So what is euthanasia? Euthanasia is also known as mercy killing in very serious health conditions where the person cannot bear the pain. Legally the person is put under a kind of mercy killing, what we call it as a euthanasia. It is also used to execute prisoners by lethal injection. Just like we have, people are hanged to death. There are countries where they use injections for prosecuting the prisoners. Now we move on to the next group of drugs called as antibiotics. Common term, right, students? You would have heard all the time antibiotics. So what are these antibiotics? They are antibacterials, of course. They are a type of antimicrobial agents used specifically against bacteria. So that should get registered in your mind, that antibiotics is only against bacteria. And they are often used in medical treatment of bacterial infections and certain parasitic infections. They do not fight infections caused by viruses or fungi or any other microorganism. There are various antibiotics available in the market with different brand names. Okay, antibiotics are usually grouped together based on their action, and each type of antibiotic only works against certain types of bacteria or parasites. And this is why different antibiotics are used to treat different types of infection. So if you go to a physician, your doctor prescribes an antibiotic depending on the type of infection that you may have, right? A single antibiotic cannot be a wonder drug for all kind of infections. That's the meaning of it, right? So the main types of antibiotics include penicillins, cephalosporins, tetracyclines, aminoglycosides, macrolides, quinolines, etc. So the classification can be based on the chemical nature of the antibiotic, or it can be based on the mode of action these antibiotics do possess. The antibiotics works on two mechanisms. Let's see. So some antibiotics works by killing the germs or killing the microorganisms. So these are called as bactericidal antibiotics. That means they completely kill the organism. Examples include penicillin, cephalosporin, fluoroquinolines like ciprofloxacin, glycopeptides, monobactams, and carbipums. And the other group of antibiotics, if we look at, they inhibit the growth of the organism. They don't kill exactly, but they stop the growth or proliferation of the bacteria or the continuous, division of the bacterial population in the body. So these are called as bacteriostatic agents. So some of the bacteriostats as examples are tetracyclines, streptomycin, sulfonylamide, macrolide, chloramphenicol, trimethoprim, etc. Fine. Okay, so we understood that we have bactericidal antibiotics and bacteriostatic. Bactericidal will kill the bacterium, while bacteriostatic will inhibit the growth of bacteria. Depending on the range of bacterial species susceptible to these antibiotics or antibacterials, they are classified as broad spectrum, intermediate spectrum, and narrow spectrum. Now, what is broad spectrum antibiotic? When a given antibiotic can act upon a range of bacteria, a whole range of bacteria, like it can be gram positive, gram negative, then we call it as broad spectrum antibiotic. If a given antibiotic is specific only to a given group of bacteria, maybe it is only active against gram positive, like penicillin, then we call it as narrow spectrum antibiotic. The intermediate spectrum have both the kind of activity, that is, to some extent they have broad spectrum activity and to some extent it has narrow spectrum activity. Let us take the classic example of this antibiotic, penicillin. I hope you can remember the story of the first antibiotic which was discovered, that was penicillin itself. It was a serendipity, students. It wasn't a discovery which was ought to made by the scientist. It was a serendipity, accidental discovery. Who did it? Alexander Fleming. Alexander Fleming was working with this bacterium, Staphylococcus aureus, and he left the plates, the petri plates, on a Christmas vacation. He just went, and when he came back, he could see that there was a fungus which was growing, which had inhibited the growth of Staphylococcus. He took out the fungus, isolated the chemical principle out of it, and that he named as penicillin because the fungus name was Penicillium notatum. And then the whole story of penicillins, and even today we have wide range of penicillins, semi-synthetic penicillins. Penicillin in fact kills the bacteria through binding of the beta-lactam ring of the DD transpeptidase, inhibiting its cross-linking activity and preventing new cell wall formation. Now we know that the bacteria is strong only with the help of its cell wall. Now what is the cell wall made up of? It is made up of peptidoglycan. The peptidoglycan is a strong cell wall because of its cross-linking. Now if this cross-linking is disconnected with the help of this penicillin, the bacterium cannot build its strong cell wall, and thereby the bacteria cannot survive, and thus the bacteria dies. Without a cell wall, a bacterial cell is vulnerable to outside water and molecular pressures and quickly dies. And so you may get a question in your mind that why penicillin won't affect the cell wall of our own cells? The answer is very simple, that human cells do not possess cell wall nor peptidoglycan, so it is specifically acting upon the bacterium which has entered our body, right? Penicillin treatment results in bacterial cell death without affecting the human cell. Let's see the structure of penicillin. This is the general structure of penicillin. I want you to appreciate the beta-lactam ring. So this is the beta-lactam ring which interferes with the cross-linking reaction of the peptidoglycan synthesis. Next antibiotic, Chloramphenicol, it is a bacteriostatic. Hope we can understand the word bacteriostatic. What is it? It inhibits the growth of bacteria. It inhibits this growth of bacteria by inhibiting the protein synthesis. You know that in biology we understand that protein synthesis is the prime most biological process. If that is prohibited or inhibited, the organism will immediately succumb to death. It prevents protein chain elongation by inhibiting the peptidyl transferase activity of the bacterial ribosome. It specifically binds to the 23S rRNA of the 50S ribosomal subunit, preventing the peptide bond formation. So what does it mean? If I take an antibiotic chloramphenicol, does it not affect my protein synthesis? It will not. Why? Because we share a difference. Prokaryotes and eukaryotes share a difference in their protein synthesis owing to the fact that the ribosome structure is different. Okay. So this is the structure of chloramphenicol. You can see the chlorine group here. This is chloramphenicol structure. We move on to streptomycin. Streptomycin is a protein synthesis inhibitor, just like chloramphenicol. It binds to the small 16S rRNA of the 30S subunit of the bacterial ribosome irreversibly. It interferes with the binding of the formyl methionine tRNA, that is transfer RNA, to the 30S subunit and thereby inhibiting the protein synthesis. This is the structure of streptomycin. You can see the structure of streptomycin here. Now, the next group of antibiotics are tetracyclines. Tetracycline is a short-acting antibiotic which inhibits the bacterial growth by inhibiting translation, that is protein synthesis. It binds to the 30S ribosomal subunit and prevents the amino acyl tRNA from binding to the A site of the ribosome, and thereby it acts as a bacteriostatic agent. Let's see the structure. Tetracycline, you can see 1, 2, 3, 4. There are 4 rings, and that's why it's called tetracycline. You can see the substituents attached to this tetracycline. So that was just a small, introduction into the world of antibiotics. In the coming classes, we'll be discussing more on the mechanisms of the other antibiotics that we need to study. Thank you, students.